Now, since we've derived the Henderson-Hasselbalch equation, let's just talk more about pH, right? Let's talk about pH. So what is pH really? It's just measuring the H plus concentration, the proton concentration of a solution, right? So we've actually developed a pH scale, and that's between 1 and 14, where 7 is neutral, and anything below 7 is an acid, and anything above 7 is a base. So let's talk about a more distinct definition though, right? So two people, Johannes Bronsted and Thomas Lowry, developed a definition for acids and bases. They simply defined an acid as a proton donor and a base as a proton acceptor. So, for example, acetic acid is CH3COOH, and that's a type of weak acid, and ammonia, NH3, which turns into ammonium, is a base which can accept a proton and turn into NH4+. So another thing is, let's talk about strong acids, right? Strong acids are ones that completely ionize, and that means that they will, for example, hydrochloric acid will completely, when put into water, turn into protons and chloride minus. So the 7 are the 7 strong acids that completely ionize. They're nitric acid, sulfuric acid, chloric acid, perchloric acid, hydrochloric acid, hydrobromic acid, and hydroiodic acid. So moving on, so why is this important? Well, weak acids can function as a buffer. Now, what is a buffer? Buffers are used to maintain the pH. For example, that's really important, as we learned in the acidosis part, that it's critical to maintain the pH at the range of 7.35 to 7.45 of the human blood, because otherwise we start going to acidosis, right? So, for example, how does a buffer work? Well, if you have acetic acid, right, the one I showed you before, and protons and then acetate, well, if you add hydrochloric acid, one of the strong acids I just showed you, it will immediately ionize into protons and chloride minus, right? So the protons, well, we know that when the pH decreases and becomes more acidic, that's when protons increase, right? An increase of protons causes the solution to become more acidic. Well, what does a weak acid do? Well, it absorbs it, right? We have acetate, which will absorb this proton that was donated by the HCl, and then therefore it will maintain the pH, which is a really vital role, for example, in maintaining our blood pH. So what does that lead us into our next subject? Well, pH can affect amino acids, right? So what are amino acids? Amino acids are simply proteins. They make up proteins, and if we want to go into the functions of proteins, they're simply, for example, catalysts, transporters, hormones, etc. And the actual physical structure of an amino acid is the alpha carbon, right? And then you have the alpha hydrogen. You have the side chain, which is variable between which differs among the 20 amino acids. You have the amino group, and you also have the carboxylic group, right? So this is all very important, and next we will go into the 20 amino acids that appear in the genetic code, right? These are really important, and the clinical application is, for example, when you have sickle cell anemia, right? So when you have sickle cell anemia, you can have a mutation from a glutamic acid to a valine. So it's simply one mutation in hemoglobin, but it causes sickle cell anemia, and it causes, for example, very painful episodes. And so it can show you the enormous effect of amino acids, and that's why they're so important to study. Another instance is Gleevec, right? So Gleevec is a drug that treats chronic myeloid leukemia, and it was actually so effective that they quickly made it into a drug, and the FDA approved it. But what happens is they noticed that some of the patients were relapsing, and that's because they actually had a threonine they had a mutation that changed threonine to leucine, which rendered them ineffective for the drug's treatment, and so they would relapse in their treatment when using Gleevec. So once again, it just shows you why it's important, and the clinical correlate of why studying amino acids is so important. So let's get a little bit more into amino acids, right? So amino acids are chiral. Now what does that mean? Well, they have a L and D isomer. And so that just means that they have two different shapes, right, that are similar. So for example, if you have your left and right hand, even though they're basically the same shape, right, and size and everything, well, you can't overlay them on top of each other, right? Because they're mirror opposites. That's exactly what these are. So they're the same, but they're also mirror opposites. And so that's important because if you created one over the other, that might hurt you just because you have the opposite type that your body doesn't recognize. So let's talk more about these individual amino acids, right? So we have glycine. So what you have to understand for amino acids is that they have both a three-letter abbreviation and a one-letter abbreviation. So for glycine, it's GLY, and then its one letter is G. So glycine is one of the most simplest amino acids. It has the traditional amino group and the carboxyl group, and for its side group, it actually just has one hydrogen, right? Because we always have that alpha hydrogen. So the simplest amino acid, and it's achiral, right? Because the definition of chirality is 4 unique groups. And so since this has 2 of the same, then you can't have chirality. So therefore, it's the only amino acid that's achiral. For alanine, you have the 3-letter of ALA and then A. So for this, just slightly more complicated, which is a CH3, a methyl group on top, instead of a hydrogen. So we've covered the first 2 amino acids. Now let's move on. We have the aliphatic side chains. They are characterized by hydrophobic side chains, right? All that means is that they have a lot of carbons. So there's 4 of them in total. We have valine, leucine, isoleucine, and methionine, right? These are the 4 aliphatic side chains. So for valine, it's VAL and then V for its 3-letter and 1-letter abbreviations. And so for the actual structure, we have the traditional 3 groups, and then on the side chain is the CH, right? And then it divides into 2 methyl groups. So how I remember it is it's just like a V, right? Because for valine, and you have a V sticking off the side chain. For leucine, you have the same thing, except when you come off here, you actually have two methyl groups, right? And so instead of directly linking 2 methyl groups here, you have a CH2, and then you have 2 methyl groups. So it has that extra carbon. And so for isoleucine, right, similar, right, you have the CH, and then you have a methyl group coming off here. You have a branch, and then a methyl group. Methionine is MET and M. And then for this one, the key thing to note is that it's a thioether. And that just means that you have a ether group, and you also have a thiol, which is just the sulfur element. So when they say that it's characterized by hydrophobic side chains, the implication is that it has a hydrophobic effect, which stabilizes the entire 3D structure of the water-soluble proteins, right? That means that it's usually clustered in the interior of proteins. Okay, so we've covered the aliphatic side chains. Now let's move on to, for example, proline. Proline is actually pretty interesting, right? So PRO and P, because it forms a ring structure, which is pretty cool. And so you have the original alpha carbon, right there. You have the hydrogen, you have the amino group, you have the carboxylic, and its side chain actually wraps around and connects back to the nitrogen, which is pretty cool. And so this ring structure actually makes it more conformationally restricted than other amino acids. So obviously, since it's in a ring structure, it's very hard to manipulate, and so therefore it has unique implications. So let's talk about the aromatic side chains, right? So we have a few of these. I remember this as PIT, PTT, phenylalanine, right? PHE and F. So I remember that because of the sound, phenyl, it makes that F sound. And so it has the CH3 and then just the benzene ring. Over here you have tyrosine, TYRY, and so I remember that because the second letter is Y, right? And so so it's the same structure, right? Everything's the same. It's just that it has a OH on the side, sticking out of the benzene, right there. And then finally, tryptophan, trip, and then W. And for this one, I just remember it as tryptophan, and you have the PHN, and you're waving a W as your fan. And so for this one, you have, this one's a little bit more complicated. You have a ring, you have a nitrogen over here, and then you have a benzene ring. So that's a little bit more complicated than the other ones. And so what's unique about the aromatic side chains is you have delocalized pi electrons that absorb UV light, right? So all these, right, these benzene rings can absorb UV light, which is a pretty interesting characteristic, and therefore you can actually use this to your advantage to differentiate them in a solution. Tyrosine and tryptophan have different UV light absorptions on the spectra, so therefore you can use that to your advantage and use that to determine how much tyrosine or how much tryptophan is in a solution. Also right here is the fact that you have, since these are largely hydrophobic because of the large amount of carbons, but the other thing is you have trip and tyrosine, right? And both of these actually have groups that can make it polar, right? Over here you have an oxygen, which could make it polar, and over here you have a nitrogen, which you can make it polar. So those are just some things to keep in mind. So let's go to the aliphatic hydroxyl groups. So just an overall structure note, the OH group of both of these make these amino acids more hydrophilic, right? Hydrophilic is to love water. So hydro is water, philic is to love. So you have serine and threonine, SER, S, and THR, T, respectively. Those are pretty straightforward. And you simply have a OH sticking off, and for threonine you have the same structure, but just a methyl sticking off that original carbon. So just remember that these make the OH group makes it more hydrophilic. So let's talk about cysteine, CUIS, pretty straightforward once again. So it's just like, come over here, serine, right? It's just like serine, except instead of a OH, it has a SH, right? And because of the SH, what's good to note is that it's actually more reactive than OH. So that's good to know. And the interesting thing about cysteine is that it can create cysteine, which is where a disulfide bridge is formed between two cysteine amino acids. And you actually have a protein called papain, which is a cysteine protease, which can break up these disulfide bridges. Okay, so a few more groups to talk about, right? We have carboxyl, the carboxyls, which is aspartate and glutamate. So aspartate, right, over here you have aspartate, you have this, you have the oxygen double bonded to the carbon and then the O minus. Over here it's the same deal, it's just that you have an extra carbon, right? And then you have this entire group. What's unique about these is that they're both negatively charged, right? Because you see over here, you can have the oxygen, which can be negatively charged. Let's talk about the basic amino acids for a second. So you have three of them. You have lysine, arginine, and histidine. This is pretty interesting because it's you'd think it'd be A, but it's ARG and then R, right? And that's just for the second letter. Lysine is also interesting because it doesn't represent the actual wording or letters. They put it as a K for the first letter abbreviation. So for lysine, you have this carbon chain all the way to nitrogen, and you also, for this one, also have this carbon chain, except here we actually have 3 nitrogens, which is pretty interesting. Over here, you also have an entire carbon ring with 2 nitrogens. So all these are unique in the fact that they have positive charges, or they can create positive charges, right? And therefore their pKa's are usually greater than 10. Also, for histidine, it has a imidazole group. That just means that it's an aromatic ring that can carry positive charges, right? Because unlike these are not rings, but this is a ring, and it can also carry a positive charge. So let's go back up here. So we covered the carboxyls, and now we can talk about the carboxamides, right? So we have asparagine and glutamine, right? So ASN, N, right, for the N, and glutamine, Q. I'm not sure how the Q came about, but just to talk about these, we have the oxygen, right, and we also have a nitrogen on the end. Once again, oxygen, nitrogen on the end. It's just over here, you have that extra carbon. The point to make about these is the fact that they don't have charges necessarily, but they are polar, right? So they're uncharged and polar. And that's the unique thing about these. So I hope I helped. I hope all of this helped and made you understand at least some intro to biochem stuff a little bit better. And thanks for watching.